Hello, my dear students. I hope you all have been doing well. From today, we are going to start our new topic, and this is about gene expression. Actually, this topic is consist on three lecture, that is lecture number 19, 20, and 21. And in this topic, we would be studying about gene expression, that is replication, transcription, and translation. So each process of gene expression would be consist on one lecture. So that is how I will complete these all three processes of gene expression in my during my three lecture from 19 to 21. So to understand about gene expression, first of all we have to understand about what is DNA. So the DNA is a macromolecule, and DNA is a macromolecule, and like other biological macromolecule that are existing in our body, like protein, carbohydrate, lipid. So like these biological macromolecule, DNA is also a macromolecule. So what is a macromolecule? A macromolecule is that one that is made up of monomeric unit. So in the case of, as we have studied before, about the carbohydrate metabolism, about the protein and lipid metabolism, all these are macromolecules. So in case of carbohydrate, so in case of for example polysaccharides, so polysaccharide are also macromolecule, and they are made up of small a monomeric unit, that is glucose moieties. So in case of protein, we have understand studied before that protein are also macromolecule that are made up of a small monomeric unit that are amino acid. So in case of DNA are also macromolecule. DNA is also macromolecule, and it is made up of nucleotide. So in my next slides, first we would be studying about the basic structure of DNA, because it is very important for you to understand about what is DNA before understanding the gene expression. And the gene expression as actually is the metabolism of DNA. As from till now, from my first lecture to till now, we have already studied about the metabolism of carbohydrate, metabolism of protein and lipid. However, now we would be studying in this topic of gene expression how DNA would be metabolized. So DNA metabolism, its DNA is its the way the DNA express its gene, that is the gene expression. So first of all, you have to understand, means keep in mind that gene expression is the metabolism of DNA, and the DNA and in the there are three processes of gene expression, two main process: transcription and translation. However, replication is the prerequisite of transcription and translation. So this gene expression would be complete in the three processes. One is translation, transcription, and replication. And to understand about the gene expression, we have to know about what is the gene and what is the DNA. So actually, DNA is a macromolecule. It is made up of polymeric unit. It is made up of simple monomeric polynuclear. It is made up of polynucleotide. So DNA is a polymer of nucleotide. And what is nucleotide? Um, a nucleotide actually is um, a means a basic unit of DNA, and it is made up of three basic part. One is the base, and one is the sugar, and three and one is the phosphate group. So if we are talking about the tri means trinucleotide, so it would be consist on three phosphate groups. So you have to just keep in mind that nucleotide is a basic unit of DNA, and DNA is a polymer of nucleotide. And nucleotide is made up of, consist on three part. One is sugar, one is ribose sugar, one is base, one of the base from four means existing base. So we will be talking about later that what are the rest means other bases of the nucleotide, but here right now you have to keep in mind that nucleotide is composed on three parts. One is base, one is ribose sugar, and one is the phosphate group. So if a nucleotide is having three phosphate groups, so we will say it as a trinucleotide. Or if a nucleotide is having two phosphate groups, so it will be dinucleotide. And if a nucleotide is means having only one phosphate group, so it will be mononucleotide. So these are the nucleotide that are existing in the um, uh, both means DNA. So and what is the DNA? DNA is the genetic material, or is the genetic material, it is the genome, means um, so genetic material means in the living organism, genetic material are of two type. One is RNA and one is DNA. So there is the difference between DNA and RNA. And what make the difference between DNA RNA? Actually the ribose sugar and the base, one of the base. So in DNA there are three type of bases, that are four type of bases, that are adenine, guanine, cytosine, and thymine. However, in RNA, you will find adenine, guanine, cytosine, and uracil. So in RNA, you will be having uracil in place of thiamine. So this is the difference between means difference of bases between DNA and RNA. And one is the another difference between RNA and DNA. RNA is having ribose sugar, and however the DNA is having deoxyribose sugar. So in other words, you can say that RNA is having the sugar, ribose sugar, that RNA will be having, it will be having two hydroxyl group at 2 prime and 3 prime. However, the ribose sugar that DNA would be having will be me say containing only one hydroxyl group that will be present at 3 prime position. However, 2 prime position there will be only hydrogen bond that would be present. So due to the absence of one hydroxyl group at 2 prime position, the ribose sugar that is found in DNA is called as deoxyribose sugar. So the nucleotide that are made up of this deoxy sugar, or you can say deoxyribose sugar, is called as deoxynucleotide. So the DNA is containing means the DNA is the polymer of deoxynucleotide. In other words, you can say, so the DNA and RNA is are both are the genetic material, means the type of genetic material. So the entire set of DNA in a cell is called as its genome. And and how means DNA means these nucleotide means involve for the formation or for the building up of the means larger macromolecule, that is DNA or RNA. So actually this DNA, what happened, that hydroxyl group of DNA, these nucleotide make join with each other via phosphodiester bond, that would be formed between hydroxyl group between the hydroxyl group of one ribose sugar and the um, um, and the phosphate of the other ribose sugar. So this is the phosphodiester bond that join two nucleotide together, and that is how means a large number of nucleotide incorporate with each other and form this means helical structure of DNA. So the DNA is having two strings of this polynucleotide that are twisted on each other. So this is the whole DNA, means a large number of polynucleotide or a large number of nucleotide in each single strand join together and form a single means string of polynucleotide. Then these two string of polynucleotide then twist on each other and form a helical structure of DNA. So but what is difference between the DNA and RNA? RNA is based on a single means chain of the nucleotide. So and RNA is having the ribose sugar, and RNA is having uracil in place of thiamine. So this was whole about the basic, that what is the DNA. But this DNA, where this DNA is found in our body or in every cell or in any living cell. So actually this DNA in prokaryote it is found in the cytoplasm, and in eukaryotes it is found in the nucleus. But question is this, the DNA is made up of a long string of polynucleotide. So how it could be possible for there's such a long polynucleotide to accommodate in a tiny cell? So actually this long polynucleotide compact on itself, um, uh, and make a compact structure that we say as chromosome. So for its compaction, protein is also help, means this protein um, compact this long polynucleotide helical structure, and then it form chromosomes. So actually chromosomes are the are made up of protein plus DNA. So like this a human being are having 46 chromosome, means 23 pair of chromosome. So and one thing more that we have to keep in mind, that when this double string twisted on each other, so they make the nucleotide means make hydrogen bonding with each other, means that found across from each other. So this is the rule, that thiamine always make the bond with adenine, and guanine always make the bond with cytosine. So wherever we found the guanine, you will found the cytosine across from it, and wherever you found the thiamine, you will find adenine across from it. So we can see here. So that is how this the two string twisted on each other, and then they compact with the protein, and then make this structure that we say chromosome. So the complete set of the chromosome that is present in each living cell is termed as its genome. So it that it is actually this genome, and that is these are actually the this DNA that make the genome of the means this is actually means the genetic material that is found in each cell of the complete means complete you can say the complete set of the chromosome. or complete set of the genetic material that is found in each human being is termed as its genome. So now question is this, now other question is we have understand what is DNA, what is nucleotide, how it's made up of. Now we have to understand, before going to understand gene expression, we have to understand first that what is the gene. Actually gene is the particular set of the nucleotide that are present on this means double helical structure. And this nucleotide um, actually having a trait, or having, you can say in other words, having a information. For example, if the gene is having few nucleotide, that is having the sequence of means nucleotide, and this sequence is means this sequence is having information that someone body color will be white. So um, uh, so the gene will be um, correspond to that protein that will give you the white color of your skin. So actually we would be studying what is the gene. Actually gene is a you can say set of nucleotide that encode for a protein. So and this gene is consist on three part, means two basic part. One is intron, or one is exon. Actually exon means gene can be consist on both, means intron or exon. What is intron? Intron are non coding region, means non coding in intron were the nucleotide sequences that would not be means encoding um, protein, means any particular type of protein, but they would assist for the gene expression. However exon always encoded with the protein, or having the information for the production of protein. So now we will be coming our means basic topic, that is what is the gene expression. Actually gene expression is the process when DNA synthesize RNA. This would be transcription, and then when RNA translate it into protein, so that would be translation, or RNA and or the protein would be synthesized from RNA. So this process would be called as translation. So actually gene expression is the transcription or translation. Transcription and translation. So transcription is when RNA would be synthesized from DNA, and translation is when protein would be synthesized from RNA. However, before both these process, there is also a prerequisite process, and without it means um, gene cannot be expressed. So this is the translation, this is the replication. This is the process of replication. So what is the replication? Actually replication is the process when DNA synthesize its copies, or we can say when DNA synthesize DNA, so it would be replication. So here we can see that what is the replication. When DNA synthesize DNA, it will be replication. When DNA synthesize RNA, it will be transcription. When RNA synthesize protein, then it will be the translation. So before going to understand about the transcription, first we would be focus on the what is the replication. So what is the replication? So replication is actually replication is the process when DNA would be synthesized its copies, or DNA synthesize DNA. So how DNA synthesize with DNA? So when we will be having miss DNA, it will be in double stranded helical structure, that and two strand of the DNA would be twisted with on each other on itself. So this molecule, when and this means a molecule of our DNA correspond to the two strand of DNA, two single strand of DNA that would be twisted on each other. So for the replication, what happened that this two strand would be separate from each other, and then on each separate would each means strand would be act as template strand, and on each template strand a new strand would be synthesized, and that is how then one means DNA molecule would be synthesized to new DNA molecule, and these two new DNA molecule would be transferred into two new daughter cell that would be um, uh, that would be means appear on the new generation or new offspring. So this is the replication. Now we will be studying for each process replication, transcription and translation, that how this replication would be takes place in prokaryote, and we will be understand that how this replication would be takes place in the eukaryotes. So actually prokaryotes, what are the prokaryotes? First, before understanding about the prokaryotic replication, we have to understand about what are means prokaryote. So prokaryotes are those means cell that are having their genetic material that is not bounded with any membrane. Means prokaryote are the those means organism that are not having any nucleus. So we can see here this E. coli is also a prokaryotic microorganism. It is a bacteria. And why it is the prokaryote? Because its gene is genome, that is the genetic material, that is the DNA, means complete set of genome, that is only one means E. coli bacteria is having only one single circular chromosome, and it is indicating its complete genome, and its genome is found in the cytoplasm without bounded with any nuclear membrane. So due to the presence of this genome without in unbounded form with the nuclear membrane, so due to this reason we say that this is the prokaryote. However, eukaryotic organisms are those organisms that have their genetic material inside the nucleus, or they all those organisms that are having means nuclear membrane around their genetic material or around their genome. So these are the eukaryotic micro means organism. So for understand to understand prokaryotic replication, you have to understand first that how much means genetic material these prokaryotic microorganism are having. So in this case, here we are having we are discussing about the replication of a bacteria, that is E. coli. So this E. coli is having right now one, or always having, Miss E. coli is always having one single circular double stranded DNA molecule. So it will be having only one single double stranded circular DNA molecule that will be closed from all the side. So it will not be open from anywhere. So it will be single circular DNA. So and it if it is a molecule of DNA, so what does it mean? It means it is having two strand, as we have understand here. So a molecule of a DNA is having two strand of two single strand of DNA that are twisted on itself. So here in the case of the E. coli, this E. coli is having one molecule of a DNA that is consist on two single circular two single strand that are twisted on each other. So and this prokaryotic DNA is having only one origin of replication. Here we say it's ori. Ori indicate that origin of replications. So what is this origin of replication? Actually, origin of replication is where is the place. Origin of replication is a place from where the replication would be start. So why the replication start from the origin of replication? Because origin of replication is having few sequences that could be easily identified by different enzyme that have to play that has to play their role, that have to play their role during the replication process. So or that have to work during the process of replication. So here in this slide we have to understand just that this prokaryotic microorganism, that is the bacteria, here in this case is E. coli. E. coli, excuse me, E. coli is having one single circular double stranded DNA, and it is also only having one origin of replication. And how this DNA would be replicated? These two stand of a DNA molecule would be separate from each other, and two new strand would be synthesized, each one on each strand. So here would be saying means discussing that how the means replication takes place on the origin of replication, and how the double stranded DNA separate its two strand, and then a new strand would be synthesized on the each template strand. So first of all, you have to understand from this picture also, whenever you will be whenever the two strand of a double stranded DNA molecule would be separate from each other and two new strand would be synthesized, so the each parent strand would be serve as a template strand, or we say that the parent strand would be is a template stand. So what is template stand? Template stand is a stand that means give the information or to the other enzyme, or to the enzyme of a replication enzyme, to means you can say to means synthesize a chain of nucleotide that would be the corresponding or correspond to the nucleotide that will be present on the template stand. So here if we go back, so we can see here in this picture what happened here, what we discuss here. So I discussed here previously that whenever if you are having a double stranded DNA, as I am it is showing here, it is a double stranded DNA, it is having two strand, and these two strand um, are the strand of a polynucleotide, and these nucleotide join with each other via phosphodiester bond, and these nucleotide join from the nucleotide that are present across from them on the other strand of the DNA via hydrogen bonding. And how they are means joining with each other? They are joining via hydrogen bonding. So if just assume that if these two strands would be separate from each other, so if you are having guanine, thiamine, and this strand would be separate from this one strand, so what would be happen then? A new strand would be synthesized on this template strand. This one strand would be act as a template strand, and a new nucleotide, a new strand of polynucleotide would be means synthesized on the template strand. So the uh, as here we are seeing here, these two when template stand would be separate, so new strand of synthesize would be new strand of nucleotide would be synthesized. So the what template strand does, actually template strand gives the information to the enzymes of replication to add up the nucleotide that will be the correspond means or means uh, that would be the corresponding nucleotide of the nucleotide that would be present on the template strand. So as I have already discussed with you that there is a rule that always whenever thymine would be present in the DNA strand, template strand, so a adenine will join with it, or if the guanine would be present in the template strand, so another cytosine would be joined with it. So how this cytosine and adenine would be adding on the this template strand, this is by the enzyme that are the enzyme of replication, and we are going to study about these enzyme in our coming slides. But you have to just at this stage, till this stage you have to just means understand this. Whenever template strand whenever the two DNA strand would be separate from each other, these two strand would be act as a template strand, and the template strand would be means act or would be means help uh, for the enzyme of replication to add up the nucleotide corresponding to the nucleotide that would be present on the template strand. So here we are seeing this the E. coli chromosome, and this is the origin of replication that we have seen here. So you can see that this was with this was the double stranded DNA. So now we are seeing here how this double stranded DNA uh, s- uh, separate or open up its two single strand, and how the new strand synthesize on each template strand. So whenever replication start, so what happened first of all, from this picture we have to understand this about the enzyme and protein that would help in replication. So for the replication there were various enzymes that would be help. So one by one we would be discussing about their name and their role that they play in the during the replication. So first of all, first enzyme that would be useful or use or play their role in the replication is the helicase enzyme. So what this helicase enzyme do, it this helicase enzyme actually is a unzipping enzyme, means it would unzip two strand of DNA. So where it will be take place, it will be takes place near the origin of replication or on the origin of replication. So when this DNA stand, like we can see here at the origin of replication, the helicase enzyme would be unzip this two strand of DNA. So first would be the first enzyme that will be involved for the means for the initiation of, you can say, replication would be helicase enzyme. And then what would be happen then after that, another enzyme that is the primase enzyme would synthesize a small primer on the template stand. So what is the purpose of this primer? Actually primer is a short piece of DNA or short nucleotide of DNA, or short piece of DNA that is consist on few nucleotide. So actually this primer would assist for DNA polymerase to start polymerization, or to start polymerizing the many or thousands of nucleotide on the template stand. So without the primer, actually without the primer it would be difficult for DNA polymerase to start the polymerization process. So The next means second enzyme would be the primase that will synthesize a short primer on the DNA template stand. Now the third enzyme would be the DNA polymerase 3, that is the main enzyme of DNA replication. And this DNA polymerase 3, what it will do, it will actually means insert the nucleotide, or it what it does, it will, we can see here. So this polymerase, what this polymerase will do, this polymerase actually will add up the new nucleotide on the DNA template stand via making phosphodiester bond between um, one rib- hydroxyl of one ribose and the phosphate of other ribose of the other nucleotide. So um, one ribose sugar of previous nucleotide would make means hydroxyl group that would be present on the one ribose sugar of the previous nucleotide would attack on alpha phosphate group, and then alpha phosphate group of the ribose sugar of the other nucleotide, and that is how it will make a phosphodiester bond between two nucleotides. So this reaction would be catalyzed by DNA polymerase, and that is how a large number of nucleotide would be add up on this template stand. So it will be done by DNA polymerase 3. And after that, would happen, after that what would happen after the means adding up a large number of nucleotide on the DNA template stand, what will happen, this DNA polymerase 1 would remove this primer, and then after the removal of this primer, the nick that will be remaining would be filled up by this DNA ligase enzyme. And then what this topoisomerase would be doing here, actually when this helicase enzyme separate two DNA strain, so the DNA would suffer from a mistension or supercoiling. So this topoisomerase enzyme would release this supercoiling, and there is also single stranded binding protein. What does this single stranded binding protein is doing, whenever this two DNA template stand would be separate from each other, so what will happen, the single stranded binding protein would bind on each template stand and make it straight. So DNA polymerase easily will run on the template stand and would synthesize the new stand on the template stand. So one thing more that have you have to understand here, that this DNA polymerase only has ability to synthesize DNA from 5 prime to 3 prime direction. So the DNA means that we are having, it is having two stand. One stand would be 5 prime to 3 prime, and other stand would be 3 to 5 prime. So on the stand that is 5 to 3 prime, we say this stand as a lagging stand, and the stand that is 3 to 5 prime, it is the leading stand. Why it is the leading stand? Because this template stand is from 5 to 3 to 5 prime. So the DNA polymerase 3 synthesize DNA always 5 to 3 prime. So it is a rule of the replication or synthesis of DNA that always DNA synthesis would be means go from 5 to 3 prime. So the stand on that, the DNA synthesis would be means complete into 5 from 5 to 3 prime direction. We say this stand as a leading stand. However, the other stand on that, the synthesis is again means take will go on 5 to 3 prime, but the stand would be 5 to 3 prime. So it would be the lagging stand. So actually, what is the problem? The DNA polymerase 3 or DNA polymerase enzyme could not synthesize DNA from 3 to 5 prime. So it can only synthesize DNA from 5 to 3 prime. So the template strand, that is the leading strand, that is having its direction from 3 to 5 prime. In this is template strand, DNA polymerase easily synthesize DNA from 5 to 3 prime. However, what is the problem? DNA polymerase um, suffers problem when it has to synthesize means DNA on the leading strand, because the leading strand is 5 to 3 prime direction, and DNA polymerase cannot run on the means on the this leading strand from 3 to 5. So how this DNA polymerase overcome this difficulty? So DNA polymerase 3 overcome this difficulty by synthesizing the DNA on the leading strand in short fragments. So these fragments that are synthesized on the leading strand in means these short fragment of DNA that are synthesized by DNA polymerase 3 on the leading strand are termed as Okazaki fragment. So the DNA polymerase 3 synthesize Okazaki fragment on the leading strand, and these Okazaki fragment are also always synthesized from 5 to 3 direction. So you have to keep in mind that DNA replication would always go, or would always means DNA replication always go from 5 to 3 prime direction, means DNA polymerase 3 always synthesize DNA from 5 to 3 prime direction. So so this was the when the two strand of DNA would be synthesized, so the two DNA molecule would be separated, and then it is transferred into two new daughter cell. So this would be the means a result of the DNA replication. So this was the old DNA replication about the how it takes place in the prokaryotes. However, now we would be studying about the DNA replication, how it would be takes place in the eukaryotes. More or less, DNA replication in prokaryotes and eukaryotes are not so much different. However, there are differences that we would study in the coming slide. But you have to understand that in the eukaryote, the DNA replication means start from the origin of replication, and the what enzyme that are taking part in the eukaryotic DNA replication. These are the single stranded binding protein. These are the protein, and what enzyme they take part in the eukaryotic DNA replication. So the first enzyme would be the helicase, that is unzip the means the two strand of the DNA on the leading is on the two strand of the DNA at the origin of replication or near the origin of replication. And then what happened, then this single stranded binding protein or SSBP, these protein would bind with the two both template strand of DNA and make them straight, means keep them straight. And then what happened after that? After that, a short primer would be synthesized on the template strand by the enzyme primase, and after that the DNA polymerase uh, uh, after that the DNA polymerase alpha would synthesize DNA would start synthesize DNA. DNA polymerase alpha would start synthesize DNA after means from this small prime RNA primer. And then what happened then? Uh, and after that what happened then? DNA polymerase delta and DNA polymerase eta then extend this DNA, and then after that what happened, then this topoisomerase enzyme would release the tension that would generate due to the uncoiling of the DNA. And after the replication, the primer would be removed by the another enzyme, DNA polymerase alpha and DNA polymerase delta. And then what happened then? And then and then this remaining gaps would be sealed by the ligase enzyme. So this was the whole about the DNA, how replication takes place in eukaryotes. So now we would be studying here that what is the difference between prokaryotic and eukaryotic replication. So first of all, we have understand about the genetic structure of DNA and RNA, that are means structure of DNA and RNA that are present in structure of the DNA that are present in prokaryote and eukaryote. So eukaryote is having the DNA that is, for example, if we will talk about the um, uh, if we will talk about the prokaryotic means microorganism or bacteria, so or the E. coli, it is having the single circular DNA. So this DNA is having only one origin of replication, so the replication would be start in the prokaryote from one point and one origin of replication. However, the eukaryote are having the DNA or their genetic material in the linear form, so it is having multiple origin of replication, and in eukaryote the replication start at multiple point. So this is one main means difference between the replication of prokaryote and eukaryote. And other difference is the as we have discussed that DNA that is present in the prokaryote are not bounded with the membrane, so the prokaryotic replication would takes place in the cytoplasm. However, eukaryotic replication would takes place in the nucleus. So it is the next difference. And one more thing, that as we know about this that um, um, uh, eukaryotic DNA um, is present in the nucleus, and the in eukaryote the DNA replication is takes place mainly during the S phase of the cell cycle. However, in the prokaryote this is a continuous process, means it is not means restricted to any phase of the cell cycle. And then there is the difference between the enzyme in prokaryote. DNA polymerase 1 and DNA polymerase 3 are involved. However, in the replication of eukaryotes, DNA polymerase alpha, eta and theta are involved. Alpha, eta and delta are involved. However, the ligases are the enzyme that are involved in the replication of both, means DNA and RNA. And also primase enzyme is also present in both, and the single stranded binding proteins are also present in both replication of both of the organisms. However, the principal enzyme of replication that are principally involved in the replication are DNA polymerases that are having difference in the prokaryotic and eukaryotic microorganism. And then in prokaryote the Okazaki fragment are large. However, the eukaryote the Okazaki fragments are small. So these are the difference between the prokaryotic and eukaryotic replication. And now we if here are these some links. So we have, if you wish to understand more in more detail, so you can go on these links. So I hope you have means find this lecture means interesting and understandable. Thank you very much, and Allah Hafiz.